to introduce our speakers today. And the goals of this group are in the Etherpad, but they're also good to say, we want to hear from the different data science programs that are being happening, also what people are teaching, how they're looking at data science funded research and what that is and what the implications of that for education are in this group. So today we'll have two talks, two Davids. So we'll have David and David. And David Beck has come before. We had another one of his colleagues at University of Washington. He's a research associate professor at University of Washington and is going to be talking to us about graduate training in data science and in the context of the university. And then David Gottman, who's at Emory here in Atlanta, kind of almost as far away as you can almost get from each other. And he is a PhD in neuroscience and an MD, and talking about their human tumor atlas and the network of data and the coordination center there. So I will let David Beck, I'll let you start. And if you want to introduce yourself again, I will stop sharing and you can go ahead and share your screen and start.
Super, thank you. Well, as much as my favorite topic is to talk about myself, I think I'll skip and go right into some of the material. Let me start my timer here. Okay, so I am in chemical engineering, as Renata mentioned, and I'm also part of the eScience Institute. This is our data science institute on the University of Washington campus. And we were formed about 2008. So if we all put our way back hats on, what everybody was talking about in 2008 was big data. So the words data science hadn't really made it into the common lexicon at that point. And all across the University of Washington campus, there were groups that were confronted with large or complex or difficult to manage data sets. And the challenge wasn't generating the data that had actually been done. What the challenge was how to extract knowledge and actionable information from those data. So we convened a group of folks from across campus and really started to think about how do we move beyond just data collection and into maximum use of those data. So we sort of broke up our task into three things: education, research, and how to build a community of practice. And so I want to talk about the education component first. Fun fact, however, this is the origin story for the logo for the eScience Institute, is where folks were coming from. So, all right. So what we decided, or what arose out of these discussions, was that there are a set of core skills that would constitute what would become data science at the University of Washington. And the first one being data management, right? If data is your currency, then managing those data is obviously an important component. In addition, statistics. So now that you have large or complex data sets, being able to ask questions of those data sets is important. And there's a statistical framework behind doing that. And it's important that folks understand concepts like multiple hypothesis testing. And so when you can test many hypotheses, that you understand how to ad hoc sort of or post hoc correct those. And so just because you discover a correlation between the number of deaths in the United States of people tangled in their bed sheets and per capita cheese consumption doesn't mean that there's any direct relationship between those two. And machine learning in all its forms is obviously very important. I'm actually going to downplay that here because I think it's overrepresented in the data science skill set. Of course, visualization. So if you have large and complex data or complex results coming out of black boxes, like some machine learning algorithms, how do you communicate those results in a visual way to your stakeholders, to policymakers, to your peers, and so forth? And then finally, one of the things that I'm really passionate about is software engineering in the scientific setting. And so we wanted to elevate a piece of software that is developed in that setting, in the academic setting, to the level of a manuscript, a patent, or a data set that you might publish. So our education goal then at the University of Washington, supported by the eScience Institute, is contextualize data science education in specific domain areas. Well, when I say specific, I really mean every domain area on campus, whether it's arts, humanities, engineering, or sciences. So we really got started with the formal program in 2013 with an IGERT. And I was the co-PI representing Chemi on that. And so we had sort of the ABCs of science and engineering here, along with CS and statistics. And the approach was to have Chemi PhDs take, for example, CSE grad level machine learning classes. We produced our first a chemical engineering PhD. The University of Washington, the first PhD was actually a chemical engineering PhD with the advanced data science option in 2015. I'm proud to say she was my student, and she's now very happily employed as a data scientist at a metabolic engineering company. Unfortunately, though, having chemi PhDs or astronomy or biology PhDs take grad level CSE machine learning classes or grad level stats statistics classes is a high prerequisite barrier. And so our participation rate is really quite low even today. So we maintain this program, and I'm happy to talk about sustainability as well. But our participation rate is about 10% of our PhD students and 0% of our master's students. And so that was another problem with that original model. So in 2016, we received an NRT. And the NRTs are the spiritual successors for the IGERT. The concept is to do more with less. So where the IGERTs were about funding trainees directly, the NRTs included some trainee funding directly, but it was more about building these sustainable programs. And all of these participating units, you can see here that they have a common theme and that they're molecularly focused. So the goal of this program was to have our students be fluent in data science methods, best practices, and tool development. So they should know how to choose a neural network architecture from a suite of available ones, how to use database query languages, how to write software using test-driven development, and do things like perform code reviews, right? So we're not turning them into computer scientists or statisticians. We're turning them into data science fluent chemical engineers or molecular engineers and so forth. Again, no prerequisites. This is super important to our program. And we wanted it accessible to our master's and PhD students. So we have it down into a format where they can complete it in 6 months, which is basically 2 quarters. We're in a quarter system at UW, and that's 3 courses. And it's a project-based approach to teaching these 5 things that I talked about. Because all the departments have this molecular focus, we can focus on teaching the students things like machine learning based on molecular properties and not predicting frenemy networks. So it's highly contextualized for them, and that provides a lot more utility, we think. At least the evaluations seem to hint at that. We leverage the cohort effect really heavily. When we were in person, we were making use of the active learning classrooms, and there's some semblance of this with Zooms and breakouts, but you can see a pod of students can work together and share their screens. Here, they can share back to the whole room, and I can project to each one of these. And so they've been really useful for this kind of teaching. Also, Jupyter notebooks have been tremendously helpful. So this is a great way for folks that have very little computing experience to skill up quickly and do it in a very safe environment that's not intimidating. We have these transcriptable options. I mentioned them briefly, but basically, if you complete this program, you get a modifier of your degree. So it'll be a master's in chemical engineering with a data science option, or a master's in material science with a data science option, or PhD as well. And we chose Python for a number of reasons I'm happy to discuss separately. So here are the 3 courses. In winter quarter, they'll take a software engineering for molecular data science class, and then a data science methods survey class. And we're actually in the process of doing a name change for this class, because although the original NRT was focused on molecular methods for clean energy, we've since moved beyond that and now include environment and health, as well as other areas. And then in the spring quarter is our capstone. So after this meeting, I'll go and I'll teach the first class of that capstone program this quarter. So these 2 courses in the winter run concurrently, which means all students take them at the same time. They spend about 8 hours a week with me and the TAs, which is, that's a pretty significant contact time investment on their end. And we use things like Slack and other kinds of communication tools to enable that cohort effect, and also study groups and a lot of peer support. When you have a no prereq program like this, you definitely get a bilateral distribution. We've observed this in a couple other settings. So you get the folks who were chemistry undergrads and minored in computer science, and then the folks who were chemie undergrads and never took any computer science classes. And so those are the kind of folks that are coming in here, and having the ability to use the digital tools for doing peer learning and peer support has been really great. And then they do one group-based project across the 2 courses. I'm going to skip over this because I'm running a little bit behind, but I'll show you a visual syllabus for how we orient the classes. So we have, again, 2 courses, the software engineering class and the data science methods class. Software engineering is in the morning twice a week, and then in the afternoon, the other class twice a week. So that's how we get our 6 hours a week. So in a normal 2-course sort of concurrent situation, you deliver content in the morning and then separately deliver content in the afternoon. Because all of the students take these same courses, that gives us a lot of flexibility to kind of front load a lot of the programming and software engineering, so that by the time they start doing the data science pieces, they have a good set of skills and a good set of software hygiene in place. So they're not producing messy notebooks, they're thinking through the software engineering and design principles while they're doing the machine learning. So again, I'm going to go through this quickly. I'm happy to share the link for this later. But we start out with the intro to programming, and you can see it's really heavily weighted during those first couple of weeks. And then we can switch over and start to balance out between the software engineering and visualization, moving to statistics. The projects start, and then they get into applied software engineering along with statistical learning, and a lot of machine learning to sort of round out the end. Because that's frankly what the students are really interested in. So afterwards, we have a big poster session. Again, this is from 2019. Usually we've done this in person, but these last couple years, it's been all virtual and digital. And it's amazing the things that the students will do when they have, the Zoom breakout rooms are dynamic, right? And they don't require a static poster. And it's amazing what the students will do with that. Move a little bit past these. So then the capstone, again, is the holistic integration of what they've done in the winter quarter. Alongside of it, we're doing soft skill preparation, so communication, project management, stakeholder engagement, really helping the students build out their research and professional networks. The capstones come from UW, national labs, NGOs, and companies. And again, our visual syllabus looks like this. So we intersperse professional development topics along with stand-ups, which are a rapid way to communicate about their progress in the software that they're building. I'm going to skip over these. Suffice it to say that almost all of these have been published. These posters that I've shown you have turned into software products that have been published. This one was shown at a couple of national meetings and was also published. So really quickly in closing, where do our folks go? So these are my students that have gone through the program. We see that there's this typical group of our master's students that go to industry or to go to PhD somewhere else. So that's sort of static and happens whether or not they participate in this. There are some that I didn't have data for when I made this figure, but this is the interesting thing, right? This is what was enabled, is that there's this whole new sort of group of folks that are going to become data scientists, molecular data scientists in a chemistry or chemistry related field. And this is new as well. We lose some. They decide they want to go work for Amazon or Google instead of staying in science. And that's great because we want optimal outcomes for the students, not just to try to keep them in science. So all of these materials are open source and available on GitHub. I'm a big believer in open source, and that also applies to the educational materials that I create. And here are some of our funders. So I'm happy to take any questions that you may have. Thank you.
So thank you, David. Yeah, I see one in the chat. Carl, did you put something in there you want to just ask?
Sure, it's actually a really simple question. Can I get a copy of that picture of your collaborative classroom? We're working on designing a space at Trinity, and I would like some more ideas to help share with our architects. Also, since you shared all the GitHub stuff, like I'll go look at the GitHub core stuff. I'm excited about that. That would have been the second question, but.
Yeah, absolutely. I can share that. I have I might even be able to get you like the business, not the business, the footprint plans and a lot more detail if you're interested in that. There was a really long design process for those active learning classrooms, but they are
right.
Wonderful.
And we're at the beginning of that process. So whatever you can share with me is awesome. And if you can't, that's okay. Oh, yeah. And Mandy just shared something else with me, presumably along the same line. So thanks.
Absolutely.
David, I'd also like to see that footprint if you can make it available. We're looking at a new business building, so having the schematics around that would be tremendous.
Yeah, I would like to.
Well, Andrew, great.
Also, with the same request here at.
Okay.
Please, yeah.
Wow. So the building, and so that, and also if you send it to us, we can send it to the group that's here.
Oh, that would be great, Renata.
So we're going to transfer the links that are in the chat into the Etherpad too, and we're we have plans to make those available, but we can just send it out as well. So if you want to do that. Also, the GitHub link that was in the last slide, would you paste that into the Etherpad too, just so that people could directly?
Already did.
Perfect. On it. You're on it. Okay, Venkata, do you want to see your hands raised?
Hi, David. This is Venkata from AMU. I'm just curious, since you are taking students into data science, maybe to PhD, right, program, and some of them graduated. So what are your thoughts on how tough it is to train them with or without prereqs in machine learning?
Mhm. I think, so having seen the students that went through this 2013 IGERT versus the students that went through the sort of more tailored education that we did in the 2016 NRT, I think, I mean, there's apples and oranges comparison there, but it's much easier to teach the students through the NRT, the program that I just described, because one, we have the cohort effects. Two, we have them for 8 hours a week effectively for an entire quarter. So it's really intensive, and you know, it's also really focused, right? So the education that they're getting is exquisitely targeted, and many of the projects that they do is their own research. So I actually think that it's much easier to do it this way. And that's, again, the philosophy at UW, to contextualize the data science education in the domain. It's much easier to do that than it is to take the folks out of the domain and put them in CSE. At least that's been sort of my observation and my experience.
I mean, are there any strategies that you got to use, for example, you know, avoid spending more time on theoretical concepts and background and all that, and just straight go to and give the necessary hands-on skills?
Yeah, I think that's a great sort of way to describe it. I mean, you do need, of course, particularly with statistics and things like bias variance trade-off and you know, some of those kinds of concepts, you have to do some motivation through theory. And in fact, that helps the students whose learning style is more sort of algorithmic. And so it's important to do that. But I think you're right that the hands-on component is something that really benefits these students, and seeing, you know, going as quickly as possible to a decision tree for computing boiling points or something like that, or predicting boiling points is something that really resonates and then traps them and gets them excited about, oh, now let's do this with a neural network, or let's do this with some other tool. And so I think you're right. It's that getting to the hands-on as quickly as possible is something that's really, really useful.
Yeah. And I see Mandy has a question in the chat. You want to ask it, Mandy? Good morning.
Sure. I was just, I'm sorry, I missed the very beginning of your presentation, David, but you know, you're really focused on any sort of major that has a molecular component. And I think it's great. My question is, how do you decide which area of application to focus on? And you know, thinking of scaling up to other areas of application, do we wind up basically duplicating our efforts? You know, we've got one set of coursework and classes for those in molecular sciences, another for social sciences, another for arts and theater, right? I mean, it's very difficult to, like you said, take someone outside of their domain and get them to take classes in, say, CS or statistics or, you know, another discipline, where it's much easier to meet them where they're at. But then we wind up meeting everyone where they're at, and that's a lot of effort.
Yeah, so that you definitely, no, that's a great point. And so you can definitely run into a scalability issue there. Ben Marwick earlier in the series and Hunter Schaefer talked about how we're doing that at the undergraduate level, how to support the diversity of programs that way. In our chat here, I posted a link for Future Rivers, which is another program that we have at the University of Washington that is similar in spirit, in that it has the data science education, but the common theme there is folks working at the urban water nexus, right? So I think that the first thing I would say is that when you can build up a critical mass within an area, then we've figured out how to do that in a way that we can replicate it again into a new area where there's a critical mass. But it is hard to bootstrap that entirely from nothing. And so there are some shared components of these programs that I didn't really talk about, facilitated by the eScience Institute. So there's some seminars and some other less formal activities that you can support from each one of these that reduce the sort of cost of scaling out to new disciplines. But you are right that if you want to support, by the way, I didn't mention it, we have 50 to 60 folks going through this program every year. If you want to support 50 or 60 folks, that takes 4 faculty, approximately 3 to 4 faculty, or 2 faculty and some TAs to do. And that's a heavy lift to do from nothing. And so I think this is where the support from the NSF really made this possible for us. And I think it would be really difficult for us to do this in a new domain, like, I don't know, ethnomusicology or something, if we didn't have support from NSF.
That's very valuable. Yeah, we
Thank
should
you.
put that as a. I want to move to our next speaker, but if there are other questions, if there's other questions I see in the chat, can you put them in the Etherpad? And David, would you mind just looking in there and answering folks if you see them in there? Perfect. So David Gottman, you want to share your screen? Do a quick intro.
Weird. Every time I share my screen, it mutes me. Feature. My name's Dave Gottman. I'm actually going to talk to you about free data you guys can probably use for your courses. So I won't go too much about my background, but I'm actually a psychiatrist, but I spend most of my time doing cancer data science and data harmonization. And that's a question that requires alcohol to explain. Oops, wrong button. So I originally, 10 years ago, got involved in something called the Cancer Genome Atlas, which basically the NCI released, half a petabyte to a petabyte of free data, essentially, for people to play with. I got excited about it and started playing with digital pathology, or essentially they can scan biopsies. And so most of my career has been building GitHub open source tools to deal with managing terabytes of giant pictures. So I am now involved in something called the Human Tumor Atlas Network, which is another NCI, National Cancer Institute project that was funded off of the Beau Biden Cancer Moonshot initiative from a couple of years. And we're now starting to slowly see some of the fruits of that investment. And so as a data scientist, my job in this network is the NCI funded 10 different research centers, 5 pre-cancer atlases and 5 human tumor atlases, and a data coordinating center. So basically all of these different universities and groups generate many millions of dollars of data. And one of the goals of the moonshot was to make that data release to the public as quickly as possible, harmonize it and clean it up. And that's really where a lot of my work, fortunately or unfortunately, focuses. I like it, so I guess it's fortunate. So there's, I'm actually just a co-investigator on this project, and we're with Dana Farber, SAGE, Memorial Sloan Kettering, and ISB in Seattle are the kind of the major hubs for that. Although I've taken on a pretty active role. So, not shockingly, the goal of the Human Tumor Atlas Network and a Cancer Moonshot is to basically bring increased focus onto figuring out what drives cancer, oncogenesis, looking for novel treatments, looking for subtypes. And the Cancer Genome Atlas, which is now probably at least a decade old, there's been a lot of important papers that came out of this. And so the idea is it's not feasible for a single center to collect hundreds of samples on breast cancer and then spend $100,000 to $200,000 of genomics technology and share that. So that's sort of what these things do. So it really is sort of a big data process. And for these Human Tumor Atlas networks, what's good and also slightly irritating, which I'll get into in a minute, is unlike some of these earlier projects where we kind of described what experiments people should do or what kind of data they should collect, this really becomes a lot bigger of a data science question, because a lot of the technologies that we're using for this big data, for the Human Tumor Atlas Network, basically don't exist yet, or they're 2 years old. So there are no data standards. There are no sample data that I've had that would have made it much easier for me to accept the data once they actually send it to us. There's no consistency of names or harmonization. So it's been a labor of love. And what's also fun about the project is, since it's a bit of a Wild West, Harvard likes to do these type of assays, and so Harvard generates a lot of these type of assays. Stanford likes to do this type of experiment, so then they generate these type of assays. And sometimes people use the same platform differently, and sometimes they use the same different platforms in the same way. So we essentially have the entire gamut of data available. And I'll get to my favorite analogy a little bit later on, but the data standards, we want this data to be publicly accessible, searchable, indexable. And also there are so many HIPAA requirements and data cleanup requirements that it makes even some of the, call it easy GitHub stuff, a giant pain in the butt. So like I can't send my friend a thumbnail of a biopsy for him to test his clinical data portal because it hasn't been released publicly yet. So there's all of these sorts of like weird hacks you need to do in order to just to sort of beep. Sorry, heard a beep. So like I said, some of the data challenges, there's an open list of assays. We're not prescriptive. People can basically, whatever they decided they like, they get to do. We have longitudinal data, which makes it even more fun because the data has to be de-identified, but we also have to be able to track these samples longitudinally. And the centers that give us the data have to do a lot of the time shifting and stuff. So that adds a fun level of complexity. We want to be able to federate across all of these different atlases. And then once we're done with the data, it then has to go into other NCI repositories, which have their own set of data standards. So not shockingly, this is kind of almost a boilerplate slide for this type of talk, but we want everything to be reusable and extensible. So we're trying to borrow as much that already existed. I'm a big fan of iterative process, especially we just keep forgetting things that should have been in there or we didn't know they should have been in there. And we have to go back and refine our data models. It's not possible for the data coordinating center to even understand what some of these new assays that people are generating for. So as we generate these like data models and RFCs to get comments on like what we should even capture about your experiment, there's a lot of back and forth between these centers, and maybe this is my psychiatry hat speaking, but getting people to do anything is like herding cats. So even if that, people are excited, like we uploaded all of our data. Okay, cool. Well, what is it? Well, we're writing a manuscript on it. We'll let you know. So anyway, that's a whole other problem. So just as an example, we're getting single cell RNA data. We're getting site-seq, metabolomics, proteomics, microbiome. Again, a lot of this is just omic jargon. But some of these technologies are like 1 or 2 years old, and I don't even know what they do. We then have electron microscopy data, which I understand very well because it's just a lot of TIFFs, but it's tens of thousands of TIFFs. So then you're talking about many, many, many terabytes of data that no one may ever look for, but we may have to pay for in terms of storage. There's all these fun fluorescent stuff. My actual specialty is really imaging related stuff. So this is the stuff I'm interested about, but I know a lot about like the rest of the stack as well. And more importantly, the metadata attributes are another small disaster because some of the metadata we require are not actually collected by the instrument that generates it. So they have to be stuck in by a human later in order for it to be processed. And guess what? Humans make mistakes. So one of the big things about the way we're approaching this is we have a single bucket, or actually we have 23 buckets, but essentially all of the centers dump their file contents and metadata, hopefully, into one of our buckets. So we have one kind of ingestion pipeline that does the cleaning, the harmonization, and the standardization, we hope. But then, with the magic of REST APIs and your other favorite APIs, we want to basically make many different ways for this data to be accessed because it's so rich, and also because, as many of you know, a genomics database is not great at visualizing images, and an image database is not great for genomics or epidemiology or whatever. So it's very important that we can expose the data and people can essentially mash things up. And this is really how I got into this whole data science thing anyway. Like I downloaded a couple terabytes of pathology data 15 years ago and started turning that into probably unfundable grants, but at least I've gotten one. So, I'm going the wrong way, sorry. So like I said, one of my areas of particular interest, obviously I don't use the machine learning jargon, but I'm particularly interested in from all of these rich medical imaging data sets, we need to get ground truth, because even though I went to medical school, when I look at these pictures, I have no idea what I'm looking at other than it's probably tissue from something. So a lot of work actually goes into getting annotations, you know, it goes even with ImageNet, right? Someone needed to know this was a cat and someone needed to know this was a dog or a wolf. You can imagine it's a little bit harder when you're trying to differentiate between lymphocyte nuclei and fibroblast nuclei. And so, you know, as we start not only getting the raw data, but also adding these additional imaging markup data on top of it so that it actually is richer and allows non-MDs who are in a data science course to build algorithms to find purple cells and tell you what they are. We need to make sure that we're kind of capturing all this information in a good way. And, you know, like I said, I'm not going to go much of deep learning, but a lot of the excitement of these big data sets, right, is if you have a big enough data sets, it's harmonized and it's clean enough, you can, you know, feed these into your pipeline, you know, pick your widget, but you can hopefully come up with new things, new observations, new understanding, new knowledge, I think, as Dave was talking about earlier, that you just, you know, obviously can't get with 20 samples. So like I said, one of the joys of big data, you know, is my data bigger than your big data. One of the way I view this, so this is our current dashboard, and I love this. So one of our favorite centers, and apparently I'm gonna make fun of Stanford right now, they've uploaded 17 terabytes of data into our bucket. They've provided no metadata. So, and I can even tell you how much this costs to store because it's fun. But essentially, I always view it as, imagine someone just went into the garage and say, here's all my photos. They're in some boxes somewhere. Can you please find my trip to Hawaii? You know, essentially, without any of the metadata being standardized, it's just a lot of garbage. And so, unfortunately, and probably most of the people on the call can appreciate this, there's a deep lack of understanding, except for maybe with the NSA, is how important metadata is, right? Just having a lot of data that's poorly annotated, poorly organized, particularly for a project where we want to make this data available for the entire universe, because that's our mandate, is very challenging and I guess keeps data scientists employed. One of the other things that is just sort of tangential, but is sort of my favorite thing, I like playing with big images. One image now is 80 gigabytes. We actually, so if you want to download the file and play with it locally, it'll take about 11 minutes, and I have very fast internet. Even more entertaining, if you don't have 100 gigs of extra storage sitting around, it also costs the NCI about $8 for you to download that sample from Amazon. So now that we're doing a lot of this big data stuff in the cloud, and we haven't figured this out yet in HTAN, because I keep fetching about how much money it's going to be if everyone just tries to download all of this free data. There's all of these other considerations related to data security, where the data is, who can access it, and who pays for it, right? Because I've done this in the past. I buy a 10 terabyte hard drive, I plug it into my lab computer, and I say, download everything I can because I want to play. If I'm our government, I guess, and now I have to pay for Amazon's egress costs, which again are not cheap at scale, you get into all of these very interesting things in terms of where, you know, doing computation in the cloud, figuring out how to give people access to your bucket in the cloud, setting up Jupyter notebooks, setting up environments, et cetera, becomes a whole other kind of a challenge. Oops, I keep clicking on the wrong thing. So yeah, like I said, I did this envelope calculation a while ago, and that worked a minute ago. Oh, well, I guess I won't show that video. So, but the thing that excites me about this is once you have the data, you can do really cool things with it, right? So these are the stuff I like looking at. You have a pathology image or something like that. Now they have these crazy assays where instead of just looking at RG and B or looking at like one marker, you can look at 80 markers from the same piece of tissue. So now you have one image, you scanned it 80 times. There's, I don't know, 50,000 to 100,000 cells on it. And each of those cells expresses some amount of 1 to 100 markers. So now you have these, not only do you have you have the visualization, which is what I like doing, but you have all these exciting clustering and data science and annotation and all these other fun things that we're hoping we can enable once we put this data in the cloud and make the tools sane enough to do it. you know, we have the other things. This is another tool where basically it's kind of cool that Chan Zuckerberg group, you can essentially drop cell data or cell expression data, and you can build all these cool plots interactively. And, you know, these are the type of things that we want to make. And I don't want to explain this thing because I don't even understand what it is, but essentially there's all these sorts of, even, you don't necessarily have to be a biologist to play with the clusters. So if you want to see how, you know, patterns of lymphocytes, which are like your white blood cells, which evade things, change across different cancers or whatever, there's different hooks that we can make to make these sorts of giant data sets accessible to the community. Then last but not least, I am a very, very small cog in a much, much bigger project. And so, you know, I don't want to pretend I do most, if not any of this myself. So just wanted to leave up with that. And then last but not least, to the people who are interested, we have not done our first data release yet. The data will be coming probably in the next 2 months where some of it's available, but there's other giant data sets available, like the Cancer Genome Atlas. Just Google that. There's a rich data set that people, you know, if you want to do data science courses on genomics, metabolomics, proteomics, blah, 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 blah. There's a ton of just free data available for people to, you know, even if you just want to do correlations with, you know, if you're white and 75 years old and male versus white, 75 years old and female, does this gene affect your outcome or something like that in any sort of cancer? So there's different ways to even, you know, filter this data out and, you know, build all sorts of cool projects, but where you have a rich enough set of de-identified and freely accessible data to, you know, putz around and do anything you find exciting or is relevant for your work. And on that, I will pause.
Thank you, thank you, David. Data. And
that's my nickname. Yes.
Yes. No, that's really interesting. And this is, you know, I want to give time for people to answer questions. And if you have links, if you can put those in the Etherpad for when your data release, or if there's a page that people should follow for where to get it going forward, that would be great. But also just saying, you know, in the future, we do want to highlight sometimes in these talks, data sets that are open for capstones or for coursework or for other things, you know, to really highlight partners that are doing this good work. So if you're...
I know this really well. I mean, this is what I spent a decade on. So the TCGA is probably even easier for people to start with, and I'll post that in the link like in 2 seconds.
Okay, yeah, I see a request to repost the Etherpad link. So for those that join later, you can add yourself, you know, sign in, and then also you can see the notes we had so far. So does anyone have a question? I have a question, but then I'll defer. So do other people have questions for David? I'm gonna ask. So I'll go first. Oh, Mandy, do you have a question?
Oh, I put it in the chat, and thank you for that presentation, David. It seems like really, really exciting work that you guys are doing. And I actually have 2 questions now. So the first one is, and I may have missed it, these are all samples coming from real cancer patients, is that right? And at what cancer centers are they at? Specific cancer centers in the US? And do you also get scans, for example, from healthy patients?
Yes and no. So the answer is, for the Cancer Genome Atlas, it worked differently. Basically, there's 20, so I built the pathology portal, so that's the one I know the best, but I think it's 27 cancer types, 500 samples per patient. Wait, not per patient, per tumor. The way that worked is they reached out to cancer centers, mostly in the US, but all over the world. And they basically said, I have a couple of brain tumors, give me $1,000, I'll send them to you, or whatever the money was. But basically the centers the data was sent centrally, and then WashU did the mRNA sequencing, Nationwide Children's did the biopathology scanning, the DNA or the chip, whatever, the mRNA chips went to Seattle. I don't really remember. But basically the way that worked is it was a hub and spoke model where the samples went to a biorepository. They did QC. We obviously track what center it came from, but then the centers ran 50,000 mRNA chips, right? One center did it all. made sense, but was also confusing, is the technology's obviously changed over time too. So originally we were using Affymetrix, and then the V2 came out, which was cheaper, and then you have to figure out how to convert the old data to the new data. And for some things, they just kept using the old chips. For some of them, they have a little bit of both. And, you know, and then for breast cancer, they have 1,000 samples because that was deemed more, they needed more data and blah, 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 blah. But the short answer is it came from all over the world. I think there's 20,000 patients plus or minus. They did most of the same assays for most of the same cancers, but like brain tumors actually started a couple years earlier because it was their pilot as well as breast. So like I said, the platforms are mostly consistent over time, but not identical. What's annoying / good, I guess, for the Human Tumor Atlas network is it flips that model. There's no central biorepository. There's a central data repository. So every center basically says, I like breast cancer. We're going to basically very deeply characterize 100 samples. And these are the experiments that we want to do. Another, so far from one of the centers in Seattle, they've only submitted data on one patient, but I think there's half a petabyte or I don't know, hundreds of gigabytes of stuff already on one person. And I figured that out when the patient identifier for every sample was the same. And I was like, wow, that's a, you know, so this one's a little bit more of a Wild West where the scientists or the individual centers are essentially driving what type and what assays are collected. But it's a deeper dive, I guess, into a couple of cancers. Does that answer your question?
Yeah, it does. Yeah, so I would imagine there are people at these centers then that have to be able to wrangle this data to send it to you or send it to the central spoke. And then I guess my question—
strangle or wrangle?
Either one. And wrangle. But how many data scientists are on your team, all these people that you have pictured here? I know you said you're just one part. So how many folks are working on the data end of this?
I'm not muted, am I?
No.
Okay, sorry. Zoom skips. So my team is small. I only have like 3 or 4 people, but again, I'm just a co-investigator. So Sage Bionetworks in Seattle, their job essentially is to host portals for big data sets. So we have there probably like 2 or we're understaffed, to be honest, but we have like 2 or 3 FTEs there doing the AWS moving data security part of it, and that's been a total disaster off the record because I keep getting Did anyone know that if someone uploads files into your bucket, you can't look at the files they put in your bucket unless you then enable another permission? I didn't, so I couldn't view half the files that were uploaded into the bucket. Anyway, so there's like 2 or 3 FTEs there. MSKCC and Memorial Sloan Kettering, they have probably 2 FTEs. They're doing more front-end visualization stuff. Dana-Farber, we have a project manager there. We have Ethan Cerami, who's the PI, but he writes a lot of widgets and code. And then we have I don't know if I want to call her an ontologist, but we have a data standards informatician. And then there's probably 2 or 3 other developers floating around in the background as well. And then as part of the contract or grant or whatever mechanism this is, the centers have to give us the data. I mean, that's the whole point of these moonshot grants. But, you know, it's like I said, it's a moving target because, and then there's ISB, sorry, the Institute for System Biology, who are also big data people. So they're kind of working on BigQuery and like, you know, less structured ways to, again, make the data available for people to, you know, basically futz around with. So probably 15 or 20 people, but obviously not everyone's full time. You know,
yeah. So a lot of people, but it could be more, and
like everything. Yeah.
Yeah, and you referenced this, but a couple of times. But there's some motivation for these centers to donate their data, right?
There is a mandate and a motivation. So what tends to happen is most of us want to share the data, but at the same time, there's so much manual labor involved in scrub. Everyone's familiar with PHI or protected health information. So here's a fun one. So once data gets visualized by me or my team, we eventually have to submit it to the imaging data commons, which is another NCI bucket. They've decided there can't be any dates anywhere in any file. Doesn't even matter what the date is. It can be, you know, the date my scanner was turned on. It's a date. Having dealt with this, anytime there's a date, we have these conversations about whether or not it's a bad date or a good date. So every freaking file that comes in, I have to write, well, automate hopefully, but write a scrubber that basically makes sure that this date is not a bad date. No, I'm sorry. I'm just going to make them 0101, whatever year it was, right? So we're like, originally we're like, we're just going to send all terabyte of data back to Stanford and make them do it. I'm like, that'll take 2 years. How, you know, so then we have to do it because it's probably faster, but then we run into provenance where now I'm changing data that someone else submitted. And so what the hell do I do with it? I'm not going to delete it, but I also don't want to spend, again, I don't really like talking about the money, but storing stuff on Amazon is really expensive, right? So if we have, I think a petabyte of storage on Amazon is like $300,000 a year. I plus or minus, it might be cheaper now, but it's not cheap, right? So if I have to duplicate a petabyte of imaging data because I got rid of 5 bits to get rid of a date, it's a lot of cash. So then you have to deal with like, well, I'll stick it in Glacier. Okay, well then we have to build a policy to put it into Glacier. But then, right, what if I screw something up? Then I have to pull a policy to get it out of Glacier. Okay, well, as soon as a file shows up in my system, I give it a new, unique ID and everything's tagged to that. Oh, but now I changed the file, so now I have to give it a new ID. You know, so you want to keep a data scientist or someone with a huge amount of paranoia or an accountant employed. There's a lot of kind of hidden mandates. Even like originally when I thought this, who got involved, I thought someone was going to give me a couple of 100 pathology images, and then, I mean, this is my main work, right? If it's, am I still sharing or I stop? Yeah. So like this is what this is what I wrote, and I'm sure it's not going to work because it's a lot. We're trying to do something live, right? Yeah. Why would anything work if I'm? Oh, there it is. It's just slow. So like this is what I wrote. I don't know, half a decade ago. I thought I would basically take 50 images, create a new slide called humantumoratlas.org.pics, and then great, I'd let people play around with the pictures. Well, yeah, I was wrong. So, you know, we have to de-identify stuff. The data formats that people are giving us don't exist yet. We have to then clean up across all of these exciting assays. Because honestly, this is kind of, you know, this was fun to look at, you know, a decade ago. This is a lot more interesting to look at now. I don't think it's going to load. My internet's being kind of slow.
One thing is, David, you know, it sounds like there's opportunities. I'm just trying to education with data cleaning or wrangling that people might to give people experience with students. It may be
I don't want to do it.
That could happen.
But Renata, then you're back to that same question of privacy issues, and someone still has to de-anonymize it and make it so that he can share it and give access, or students have to sign non-disclosure agreement.
I think that's probably going to be his work, but in the sense that once you get it to a place you can share, there's still a lot of issue.
Oh yeah, there's a lot of stuff that can still be done. I don't, you know, students want to play with data. That's not a problem. So like what I'm doing, unfortunately, for our first data release is I'm just eating it and manually going through the first 300 images myself and looking for the tags because it's not that hard for me to do. I'm not doing it for the other 50,000 images. You know, like there's sort of what we're going to share publicly and what, and actually there's Google has tools to do this, whatever. But, you know, going back to what David said a lot is I'm not a software engineer. My dad was, and I mean, I'm weird because I'm a psychiatrist who spends most of his time coding. So I'm a very bad software engineer. But there are ways to do this well, right? You know, data goes in, scan metadata, look for scary stuff, send it to some service. It gives you some things back. Okay, then we have to write our business rules. So like, here's a fun one. I hate XML. XMLs have schemas. A lot of those schemas have dates. My tool was detecting that the schema, which I can't ever change because it's a schema, has a date in it. Now I have to write a business logic to say, this is a date, but this is a good date. Okay, now it flags another date. Now Google said, oh no, this is another scary date. I'm like, that's, you know, so there's just a lot of low-hanging fruit.
I would also say, this is just a plug for some of our groups where we have the cyber infrastructure group. There's a project they're working on for open data, open storage network for high impact scientific data reuse. So now it's live as of 2021. It's through there's 4 data hubs and then some other partners. And now it's live on Exceed. So you can request storage allocations and you can also buy your own pods. So you mentioned Amazon a lot. It's kind of a scientific alternative to Amazon. And so we can put some of those links there too. They're having a webinar later in April.
Yeah, that would be interesting to look at. The way my backend actually works is I can actually connect to an S3 or a Google bucket, right? But again, for like this image that I think I'm looking at is 70 or 80 gigs, right? I'm not pulling all 80 gigs in real time because that doesn't work very well. So my tool goes in and does magic, blah, blah. But basically, if the performance on the stuff that's on Google versus on the stuff that's local or on S3 or whatever, it's noticeably slower. I don't know how much noticeably slower it is because it's a pain to characterize, but depending on what the application is, it gets, the farther you are away from the data, even if that doesn't cost anything, the latency can go to hell. Or there's weird firewall issues, or, again, I don't want to go too crazy in the software engineering, but.
So let me spin a slightly different question. Sort of thinking off of what Renata was saying about this, lots of tasks for students to do work and things like this. And, one of the things that I always liked to do for my students when I teach these intro classes and stuff like that is I want their homework to have an impact if I can. The question, I guess, that part of this thought is as some of this data becomes openly accessible, you were saying, you're manually tagging the first 300 data set points, but the rest are all just going to be untagged or whatever. Is that process that you're using to manually tag something that can be articulated enough for students of less knowledge, but more time to do, such that you would then be so if I give a freshman a list of tasks that they have to go in and check based on your, this is what I do to tag 300 images, here's a to-do list, you have to do it for all that we, is that something that's a feasible, remember that this is a learning experience for the students. So you're not necessarily getting students that are going to be able to identify issues per se, but we want them to get experience seeing what the problems are.
Oh, yeah. So, I mean, if I have another small grant where we're working on de-identification tools. There's already data in the TCGA that has identifiers in it, and you don't have to go to medical school to see someone's name and be like, that's some dude's name, or to see, or this is a date. So a lot of the times when I try to task students, I went to doctor school at some point. I don't know what the hell I'm looking at. I guess it's a WD76845. I don't know, right? And it has a bunch of colors, and I think CD4 is an antibody for the immune system. So there's something going on. I don't know what I'm looking at, right? But when I look at it at the same time, I think I just saw a weird blue dot, right? And I'll be like, that's probably an artifact, right? Someone drew with pen, or this is crap, right? I don't know what it is, right? So there are ways you can basically define the tools that as long as someone has eyes, they don't know what it is, but they know it shouldn't be there, right? So even for we'll call it a de-identification pipeline, essentially they just have to write tools. They flag something, and then I don't know if everyone knows what regular expressions are, right? But you write a date module, you go, oh, I found a date when I was going through this before I said okay. Well, now I want to see if I can write something that will automatically find this date, right? This date format. And then you do it again, and you do it again, and you do it again. So basically, most of my life is iterative refinement of crappy code I've written, right? So yeah, there are ways definitely to, especially even for this project where we're just looking for, call them latent dates. Yeah, that's definitely something that's outsourceable.
We're at 12:01, so if anybody has to leave, you can go ahead. We'll stick around for another couple of minutes if people have questions. But if you do have questions and you have to go, put them in the Etherpad, and I think David have said that they're willing to look at those if we're going. So I just want to keep content of time. But yeah, if people have questions, feel free.
Yeah, I mean, David, that sounds like things that are certainly feasible in small bites to students of useful skill sets, right? Like, I mean, if you're having to do it as part of your job, teaching students, giving students a practice chance to do it would make sense.
If students want a summer job helping me look for dates, because what's nice about this is it's this stupid intermediate where it's probably really not PHI, right? In fact, I know it's really not something we're scared about, but we have a policy that the person who is going to get the data after me, we don't want to have this stupid conversation about dates ever again, right? Because every time you see one, we're now getting into really a cybersecurity problem where we're saying, oh crap, they put an absolute date, but we have relevant dates. So if I'm smart enough and I have enough data, can I use this one date that probably occurred around when they were at the surgery plus their age? You know, it's sort of gotten to the point where it's annoying me where, you know, this is for NCI, so we can't screw up and release data. But at the same time, the idea that there's perfect de-identification is a bit of bullshit unless you spend more money than we have. And B, I'm pretty sure someone on this call can tell me if you have enough data, you can identify anybody, right? I look at their death record. They had a glioblastoma. They lived in Boston. Okay, that was Jane Smith. I don't know, so, but again, you know, there's, you know, the date thing is a good example of something where, you know, we need to build a workflow, but it's, call it shareable, because the stuff that would be exposed is very low risk. What's funny about it, though, is I can guarantee someone's already done it. The electron microscopy data we're getting, it's really big. It's like many terabytes. There's probably dates and everything. Do I want them to re-upload it? I can't make a dog sit, as I say sometimes, right? So I don't even know if they will. They'll say, I don't think it's a date. I don't think it's important. Okay, well, I can't make you re-upload. I don't know. Do I delete it all? Do I just run a script and do it myself? What do I do with the old data? Right? I made it clear on every call. I'm not stupid enough to be the final source of provenance for any crappy script I write to get rid of anything. Right? Anytime you touch someone's data, it becomes your data. So, or I might have to rerun the script, or I learn something new, or, you know, whatever the process is. So you get into all these like data governance issues that are both interesting and unsolvable by a co-investigator on a much bigger project, right? But the real blockers, right? It's not a technical problem for me to delete half a petabyte of data, but I usually pay my taxes, and it would piss me off to give Amazon an extra $200,000 for just literally you change 4 bytes, right? I mean, if it's my birthday is 050975. Don't steal my social security stuff. If I make it 010175, right? I've now changed 05. I've changed 2 bytes, right? The 5 becomes a 1 and the 9 becomes a 1. I'm going to keep the year. Change 2 bytes. That imaging file is 80 gigabytes. It'll probably even have a new MD5 sum, I hope, if the algorithm works, right? So I've now created a new file. Do I keep both copies? Probably. Carl seems skeptical.
Well, no, I'm pondering this idea of the fact that you have metadata, right? Like part of the challenge here is that what you're saying is you're changing the metadata. Could you?
I'm changing embedded metadata in the file. The CSV stuff's cheap. Like a JSON file or a CSV file we change on the fly. But some of the stuff, we'll call it by definition, is either burnt, like a PNG or a TIFF, right? You get any picture, they put, like, if from a camera, these are magical cameras, but a normal camera, it actually can put your GPS coordinates in there. That's a bad one.
Right.
It puts the date you took the picture. I don't think it does, but it may take, it was a Samsung Galaxy S7 200. I don't know.
Yeah. You know, so it's stuff that's embedded in the actual image file. And so then the question then becomes, should you remove all of the embedded data and only store the image, quote, image data, and then connect all your metadata via elsewhere? Or are you, I mean, the sort of that never mess with your raw data question. And you're right, like that's sort of I beat that into my students, right? Like never modify your raw data, but if you don't, if your raw data is 80 terabytes, do you store a second copy?
Yeah, well, that's the issue. It's also to a certain extent above my pay grade, because technically the data should be de-identified before it comes to us, but it's never going to perfectly happen. And again, just pragmatically, I have very fast internet, but to re-upload half a petabyte of EM data, it'd take 4 weeks, right? If it's already on S3, it doesn't, I think. I don't know. Or what, you get into all these like pragmatic, whatever you want to call it, where like there's latent costs and everything, right? So again, the biggest example is I did this for TCGA. I downloaded all 20 terabytes of data a lot, right? Now, some of the data we're talking about is even bigger than that. And, I have a NAS at home, but I don't have that much space. It's fun. These are fun things to think about. What also happens is, so Carl, the other problem though is then instead of all of the data I need to look at an image being in a bucket that the NCI owns, this metadata is now stored in some other magical database that if you're not, probably some of the people on the call, you don't know how to write the Python REST API script to grab. In fact, I mentioned this. This is what I wanted to do initially is you download the bits, you then have a magical renaming script that basically pulls the metadata you want, puts it in the file names, and makes it not a total pain in the ass to work with, right? Didn't go over very well, right? And we even have this stuff with semantics names, right? So essentially, my stupid system basically stores everything on my file system just using MD5 sums. So it's EA7767. I'm looking for Gottman dash not tumor healthy guy in the world dot picture, right? I have to look up the MD5 sum of that original file or whatever. But then we have this other problem is as soon as I call it Gottman's normal healthy tissue dot whatever, I've now embedded semantic information in the label. And then it turns out Gottman wasn't so healthy because he has COVID. So now my file name, which I was using to have semantic meaning, is now bullshit because later on it turned out I did it wrong. So we actually had this happen with the Cancer Genome Atlas where, they didn't actually have a brain tumor. It was a breast tumor. Or I'm kidding, but you know what I mean? Like where you update something, just because you have the bits doesn't mean whatever you've associated with are correct. And then you get into versioning and data and, who's in the know to know that, I should have re-downloaded the data because, Carl didn't have breast cancer. It was a prostate problem or what, sorry, Carl, you're the first one who shows up on my list. Yeah.
No, I mean,
it was Mandy's prostate.
Yeah.
That's fun. I like thinking about this stuff.
Because, the data, the importance of metadata, how to also get this across. I think this is so important, and I think it's lost sometimes in the idea of trying to make things simple for people coming into fields where you think, okay, you won't spend years on these types of problems, but these are really kind of fundamental blockers to some of the things
80% of
do in research and like being able to be good at this. There are practices, there are, working groups. I know that, when I was in government, I went to all these working groups for DARPA where they had this exact same problem. So there's people that have been thinking about these things, but yet the propagation of it down.
So switching topics briefly. So at the VA, I pretend I'm a psychiatrist, but I mostly do data stuff now because I know how to program. So like we now have thousands of people we called to schedule the vaccine. They actually got it somewhere else, but we now have a flag somewhere that says they got it somewhere else, but it doesn't show up on our numbers because it has to be documented in some other system. So now we have to have nurses call back 3,000 people and then change another bit in some other table so that this dashboard gets updated nationally so we get credit and blah, blah, blah. So yeah, and then it's a data science problem because I know where to find the stuff, but calling 3,000 people is not my problem. So like, you can come up with even like, we'll call them quote unquote simple data science problems that are not simple. And even when I say, hey, there's 3,000 people you guys need to call, I don't know if that's an actionable problem. Do we have the staff to call it? Or do I have the money? What if I just didn't have the money to store both copies of the data, right?
Mhm.
My 500 petabytes of garbage. Do I delete it? I'm not making that decision. Let Mandy make it. She
Mhm.
pays taxes too. Or Derek. I don't know, whoever wants to pay taxes on it.
Yeah, we need the astrophysics people and we need the medical people. They know how to get rid of data real time. The astrophysics people do, at least. But then their data doesn't have any PHI, so they don't have to de-identify anything. So they have an advantage and a disadvantage.
Well, and
yeah, they can.
It also makes me wonder if like part of the challenge needs to be, you know, if you think about how Git large file storage works, right? Like you're only ever uploading the differences, and then, but the problem is that if you have like a difference file that's tracking, somehow you can reverse, and you know, someone could reverse engineer all the data that you've removed out if all you've done is track a difference of saying I've removed out these bytes. But it's also a much more affordable thing than storing a second raw copy. And so there's a whole bunch of like governance. There's a whole bunch of like just raw practical, like how do we manage this data things to be addressed.
And that is a big, so the way AWS works, or at least the buckets, right? A bucket, once it's there, the object's immutable from what I can understand. So, well, I know that because apparently in order to give me permission to these files that I couldn't read, we actually have to copy them to the same bucket, but apply a different policy. So they said it's going to take 200 hours. Right. So like, you know, it also depends, and this gets into the nuance that probably only three of us care about, right? But there's like block level storage versus object storage versus SSDs and NVMe and all these sorts of things that, you know, a lot of this is supposed to be so abstracted, but then you actually like, I'm like, the performance sucks, guys. Well, maybe Carl will appreciate this, but we put off all of our data and they just mounted it with an S3 Fuse file system. And not surprisingly, the performance is terrible compared to it being on a SSD drive in the same server that it used to be on. And they're like, everything's slow now, guys. I go, yeah, because you did something stupid that was not going to work, but you saved money. I don't know. Like, we ran out of disk space.
They saved a lot of money because an SSD is way more expensive than a standard drive. And
yeah.
So if you have, you know, right, like there's scales. The faster you go, the more expensive it gets. Like, cache is super fast, RAM is super fast, but it's also super expensive for gigabytes of data. So
everything's stored in the CPU registers. Yeah. Anyway.
You know, it's well, I mean, the thing is, it's a neat engineering problem also that sort of circles back around to that. Now that we're getting more advanced in how we do the technology top use, right? There's now these problems that have to get pushed back down to hardware engineering problems that say we have problems that, like, because we are managing data of this size, scope, complexity that needs to be connected, we now have completely different like architecture needs for how do we manage and use it. And in some sense, the computational mechanisms and the hardware mechanisms to deal with it have not caught up with the fact that we have these issues, right? Because you could imagine, okay, I can imagine some completely alternatively different ways of having your data stored in hardware. And so like some of the like Haskell and stuff like that actually access the data bits differently to improve the processing speed. Because
we've done that actually. We did something similar where if we change the way the bits are organized in our images, the read speed goes up by 30%.
Exactly, because I watched it. There's this fantastic video that I used for my computational linear algebra class that talks about like the different ways in which you scan the data depending upon the kinds of operations you want to do. And so when you're trying to do certain kinds of masking or certain sorts of convolutions and things like that, you don't want to use a standard data read that reads all of the column of data of bits, because you actually you need a different locality. And the problem is that, well, the issue with that is that until we reorganize how our hardware stores the bits and data, even when we reorganize our computation to improve locality on the computation, we haven't actually improved the storage access to it. So all we're doing is fixing the really, really fast memory usage, but we're not fixing the access to the fast usage, where if we were storing those image files differently on the hard drive itself, then we would actually be able to improve read, write, compute access, because the whole pipeline would be faster.
You can teach that though very easily with, you ever use pandas and do it wrong?
Yeah.
Sorry, we can. Yeah, Venkata has had their hand up for a while.
Yeah. So go ahead. Someone has, if you wanted to talk, we have about 2 more minutes, 2 to 5 more minutes. And then I put in the link just for you, the openstorage.network.org. You know, I know you already have a setup with AWS, but because of the, it's new, it went live. It's like a distributed system of pods at different universities that are leveraging NSF cyber infrastructure, and is now part of Exceed. But they have, from what I can tell, they try to use the same protocols like the S3 and other things to be able to link similarly to Amazon. So
Yeah, I'll check it out. Thanks.
Yeah, just to see how it goes. And all right, so does anyone have any final things? I know we probably 12:20 have to close it down, but
yeah, it's not final thing. I just had a quick question.
Sure.
So, you know, it's all nice discussion. Actually, it's going to help. But I was talking about, I was about to ask the date issue in the data. Is it mandated that there should not be any date, or only the birth date or similar things?
So originally, this is why it's not a PHI problem, it's a policy problem. So yes, originally we wrote our policies worried about PHI, but then one of the upstream places that will eventually be the home of all of the data basically does not want any date anywhere in any file. So it's more of a policy of the called the upstream host, which again, it's having had these conversations for over a decade where you keep wondering whether or not this date can be connected.
Yeah. Is it because they could not interpret correctly the HIPAA, or they're scared of HIPAA, or what is the reason behind just scrapping all the dates? Because sometimes you might need some dates especially for a longitudinal study, right? Like the example you were giving, only one sample of that person, that many petabytes of pictures, meaning it's not taken all on one date, right? So
no, we're requiring them to date shift and then use relative dates or something, or like days since diagnosis or whatever. So I agree with you. It's a bit of an overstatement, but at the same time, having even dealt with IRBs, where I got in a fight like 10 years ago, where I just wrote my birth date and sodium 144. Right? People basically, the people interpreting these policies sometimes just say a date, the word date is in HIPAA. And then we started getting into these philosophical conversations. If I knew what month someone's surgery was in, and I knew the relative date from some other data sources, could I use an absolute date and a relative date to do something out? It started getting esoteric, but at the same time, these are big money initiatives. And if you've ever dealt with a data breach, it sucks. So, one way to do it is just say, hey, if it looks like a date and talks like a date, it's a PHI date. And I don't agree with it, but that's sort of what came down the pipeline.
Yeah. Well, because if there is a mandate like HIPAA, we have to make sure we follow it unless it is modified further. But I think all these examples and issues and, the importance of having some dates for longitudinal research should go into that, that policy meeting and slowly change it while balancing, the act of maintaining the privacy. You can do both, I guess, but it takes time to make them understand.
Yeah.
That's all I want to say. Yeah. I don't want to take any more time.
Yep.
Yeah. Well, thank you.
Thank
This
you.
has been great. A great session. I always find these to be engaging. So we'll connect next month for sure. And you come back, David. Well, always welcome.
Thanks. Thanks again.
All right. Thanks again. Bye.
Renata, could you